So it will be marking the class test paper. So please go through the answers and check your answer. So part B, the rate of ATP consumption of demyelinated neuron is greater than that of a myelinated neuron when conducting impulses at the same frequency. So explain why. So that means to say for a non-myelinated neuron, the entire stretch of the membrane of the neuron, they need the sodium-potassium pump, right, to create the resting potential. So the whole neuron, whereas in the myelinated neuron, only at the node of Ranvier that they will need the sodium-potassium pump to pump the sodium ions. Okay, so let's look at the mark scheme. So action potential only travel along the membrane of the axon. Sorry, action potential travels continuously along the membrane of axon in the demyelinated neuron versus action potential jumping from node to node, which is called saltatory, not salutatory. Saltatory conduction from node to node in myelinated neuron. So because of that, there'll be a longer length of membrane for depolarization, repolarization, hyperpolarization, action potential, electrical potential. So this one is in non-myelinated neuron. So longer length of membrane for action potential to travel. Longer length of depolarization is needed. So they need more sodium-potassium pump to restore the resting potential. So if this is an axon, right, the entire axon, the membrane will need to depolarize. Whereas if another axon has got the myelin sheath around it, which is the red color one, this myelin sheath, right, covering almost all the membrane. So the length where depolarization occur will only be here, okay, and here, which is node of Ranvier. So this is where depolarization will occur for action to potential to be generated. So only sodium potassium pump is needed here. So less sodium potassium pump. Okay. Okay. Everyone okay? Okay, next question. Part C. Am I recording? Curious. Yes, I'm recording. Okay. Part C. I'll give you the mark scheme. You want the mark scheme, right? I'll give you the mark scheme, okay? Okay, figure 1.1 shows changes in pre-synatic and post-synatic membrane potential following the arrival of single impulse at the synapse. So this is pre-synatic membrane. So the impulse already arrived at timing maybe about 3.5, about 3 to 3.5 millisecond. So this is post-synatic membrane. So what does the question ask you to do? In the figure, show the post-synatic membrane potential following arrival of the single impulse at the synapse, whereby the intensity of the stimulus at the receptor is too low to be known by this person. So that means to say, you see here, they label this pre-synatic membrane potential. That means this is the synapse, right? Action potential arrived to synapse already. But then they said this action potential stimulus is too low for the person to actually to be able to know it. That means to say that no action potential be sent, isn't it? But is there a local depolarization? There's a stimulus, right? Whenever there's stimulus to the neuron, it will activate depolarization, isn't it? So there are three kinds of stimulus an axon can receive. Either it is electrical, like this one here is electrical, or chemical, or mechanical. So these three stimulus arrive to the axon, can stimulate axon to depolarize. So what will be the answer is, you can, first is the timing. The timing after they receive the, make it smaller. Huh? So the timing after they receive the action potential, it must be after that. Then you can draw anywhere starting from resting potential, right? So it must be at rest first. So initially must be 70. Then after any timing after the peak of this action potential, you can draw, it will go up, but it will come down. That means there must be a certain location where this is a threshold. This is a threshold potential. So this one doesn't go above the threshold. That's why it comes back down to resting potential again. Okay? So what is the mark scheme? Start after the peak of the presynaptic membrane potential. Start anywhere after the peak. This is when it has received the stimulus. And then the peak must be below the threshold potential, -20 millivolt. And then what else? Recovery back to resting potential. So starting point must be after the peak at the presynaptic membrane. So it starts after the peak. Right, this is the peak. So it starts after the peak, but it doesn't go above -20 millivolt. So -20 is around here, which is the threshold potential. And after that, it will slowly go back to the resting potential. So these are the 3 marking points. Okay. So question 2. Question 2 is actually the textbook question. Question 2. So the diagram shows a sarcomere, which is a unit of contraction in the striated muscle, in different states of contraction. So A, B, C, and D. So A is name the parts labeled P, Q, and R. Okay, so P is myosin, the thick filament. Q is actin, or the thin filament. R is the Z line, or the Z disc. Okay, PQR. P, actin. Sorry, P is the myosin. Q, I think they know lah. It's wrong lah. Q is the Z line. Q is the Z line or the disc, and R is the actin. Okay, actin. Okay, so next question. B, explain why there are no actin-myosin cross bridges visible in diagram A. So A, you can see that there's no cross bridges like this one, right? The V shape. So here don't have means what? The muscles are not contracting, isn't it? Right? There's no muscle contraction. So 2 marks. How to explain this one? Because the muscle is relaxed. Muscle is relaxed. The troponin and tropomyosin molecules are covering the myosin binding sites on the actin filament. So there are 2 molecules, troponin and tropomyosin, that are covering the myosin binding site on the actin filament. So this is a full description. So therefore, myosin cannot bind to the actin. Myosin head, right, cannot bind to the actin to form the cross bridges. So C, muscle fibers are able to contract with more force in some states of contraction than the others. So that means some contract stronger, some contract less strong. Suggest which of the diagram shows a state and that can develop the greatest force, and explain your answer. 4 marks. So you know that among all the 4, diagram, definitely D is the one that give the most force, isn't it? Because it's pulling many times in order to pull the entire actin into the, pulling entire myosin inwards, right? Okay, pulling the entire myosin inwards. So how to write answer to score for this one? So answer is C. Hang on, C, D. Answer is D, right? This is from a textbook answer directly. So I need to modify this part here. So answer is D, right? Answer is D. Because D has got the greatest overlap between the thick and the thin filaments. D because greatest overlap. Greatest overlap means there is maximum number of cross bridges between the thick and thin filament, or between the myosin and actin. So you know that every time a cross link is being made, there will be a power stroke, isn't it? Every time a cross link is made between the myosin head and actin, immediately there'll be a power stroke. So the more cross links you have, therefore, I would say there'll be more power strokes. I'll write there's more power stroke. So therefore, more power stroke means greatest force. You have the greatest force. You have the most number of myosin head that is pulling the actin inwards. So therefore, there'll be the greatest force. So I would say that you've got the more number of myosin head that create the power stroke. Yes. So there'll be more pulling of actin filaments towards M line. Okay. No, you see, they say C is the answer, not D, you know. They say in D, they cannot pull anymore. Okay. Okay, I think what they say is correct. You see, D already finished already. D already finished already. You cannot pull anymore, whereas C can still pull. D cannot, because they said subcranial cannot shorten anymore. That's why they said answer is C. So answer is C. Answer is C. Yeah. Answer is C. Not D, Yes. Yes. D. Huh? Why not B? Okay, let's see why not B. Because B, the number of crosslink is less than C. That's why. No, the thing is because in D, they say that D already is like your actin already, both the actin already arrived to the M line. It can no longer move anymore. So you cannot create any force to pull the muscle anymore in D. Again, D, you cannot, yes, there's maximum crosslink, but it cannot do anything because already all your actin already touching the M line already, so it cannot pull anymore. Whereas compared to B and C, anytime we have got more crosslinks than B, you know. So the more crosslinks you have, the stronger will be the power stroke. The more crosslink, that means the more myosin head will create the power stroke, the stronger will be the force in C compared to B. So therefore, wow, this is very tricky. How do I know that they arrived there? You see this one move until like here is that touching. Can you see not? This line, the gray color line is touching the black color line, which is the end disc. So you already banged the wall already, cannot move anymore. Okay, so wow, very good question. So answer is C. These are, textbook questions are really good. Miss Annie got ask you to do or not? Textbook question. Hey, I could give you mark scheme. So textbook questions are very good. That's why I use textbook questions. Good, man. Good. That's why I use. No good, no use. Okay. Right. Question 3. A finish already. C finish already. Oh, finish. Okay, now kidney. Kidney question very easy. I give you simple, straightforward. Correct or not? Okay, label structure A, B, C, E, F, G on the diagram. Half mark for each correct label. Spelling wrong, no mark. Okay, please check your answer. Explain why the fluid concentration in G may remain the same as in F. Where's G and F? G is in the collecting duct, same as in the distal convoluted tubule. So if the concentration is the same, is there a reabsorption in G? No, right? You didn't change anything inside the renal fluid flowing in G. That's why there's no change in concentration. So why is there no change? Let's look at the mark schema. There's my mark schema. Yeah, okay. 2 marks, right? Because ADH is not secreted. Why is it not secreted? Because when the blood water content is high, so ADH is not secreted. So both DCT and collecting duct will not be permeable to water, so water will not be reabsorbed. Do you know that both DCT and collecting duct can be regulated by ADH? They'll be regulated together, right? So that's why no reabsorption because blood water content is high. Okay. So since when I stopped recording? Okay, this is how it is, like something like this lah. Somewhere here. Cannot. You know that loop of Henle is the one in the medulla, right? Hello? You need to know location. No, this is what? This cortex, isn't it? Cortex, and this is medulla, right? So whatever cells it is here, they're all immersed in the tissue fluid surrounding them, right? So if you're talking about loop of Henle and collecting duct, then it'll be tissue fluid surrounded by tissue fluid in medulla. Whereas if you see Bowman's capsule or renal capsule and then PCT, DCT, and a little bit of the beginning part of collecting duct will be located in cortex. So surrounded by tissue fluid in cortex. So when you say reabsorption of materials like water, sodium ions, like over here, right? Not over here, the PCT, proximal convoluted tubule, there's a selective reabsorption, right? So the things are reabsorbed into where you already know what? They pass through the wall, isn't it, of the PCT, right? The epithelial cell of PCT, go through the basement membrane, then go to surrounding capillary, isn't it? So what is the name of the capillary? This one that surrounds here. Peritubular capillary. So you need to remember. Peritubular capillary. Peri means perimeter, means surrounding. Tubular means the tube lah, PCT, DCT. Whereas there's also the group of capillary over here, right? Where does it come from? So it must have connected to here as well. So this group capillary is called vasa recta. So you also have to remember, this is where we reabsorb material, vasa recta. We reabsorb material back into our bodies, have to back into this blood vessel, then go back to the body through the bloodstream. Okay? So the directions were from inside here. So let's say water reabsorb first into the tissue fluid in medulla. Then after that, go into the bloodstream in vasa recta. Right? Okay? Right. So next question. This one is to ask you to label, right? Label where? Indicate clearly in figure where ultrafiltration occurs and selective reabsorption occur. Ultrafiltration occur must be between the capillary, the glomerulus capillary, with the inner wall of the Bowman's capsule, which is anywhere around here. This entire mass, as long as you put anywhere inside this region, is correct already. Okay, not only the side, not this side, it's actually the mass inside here. Okay, so it's ultrafiltration. Number one. Number two, selective reabsorption occurs in the PCT. You know that PCT has a lot of microvilli, right? So therefore, if you look at the lumen, right, the border between the space in the lumen and the cell is a bit a shadow one. It's a shadow one. Whereas you can see this one is borderline is very clear, right? Between the lumen and the cell. This borderline is got shadow because of the microvilli. So therefore, you label anywhere. This one is correct. This one is correct. This one is also correct. Okay. Others are not correct. Do you know what is this one here? DCT, yeah? DCT, yeah? Okay. Because this photograph is taken in the cortex of the kidney. Right? Cortex, you only contain either PCT, DCT, and renal capsule. Okay. And a little bit of collecting duct. You know how to see collecting duct, right? The borderline between the cells are very clear one. You know, right? Okay. Okay. Okay, the graph. Question. Explain why sodium ion concentration increases in region C and D. So this is C and D. Why the sodium ion concentration increase? That means there's reabsorption of water, right? Reabsorption of water in C, which is the DCT. D, which is collecting duct. So let's look at the mark scheme. Water is reabsorbed when ADH is present. Or when low blood water content.
Secretion.
Secretion of what? DCT function is no longer in syllabus, so I didn't teach my class. DCT function you all got learned or not? SPM.
Yeah.
SPM, okay. Now what? Nowadays learn less thing. Okay. Question 4 is essay, right? Is it essay? Question 4 is essay, You take all the essay question and then you convert to 5 marks, will be your current structure question. Okay? Take all the past year essay, convert to 5 mark question, it becomes your structure question. Right? So, number 4, describe the main stages of cell signaling in the control of blood glucose concentration when by adrenaline is similar as by glucagon. This one is similar as glucagon, okay? That means your body needs a lot of glucose at that time. When you got adrenaline or when you glucagon, your body needs glucose. So we need to mobilize the glucose. So this is the past year, November 16. So adrenaline binds to the receptor in the cell surface membrane. Then the receptor will changes conformation, means changes shape. The shape of protein is also called conformation, so changes conformation. G protein is activated. It will cause adenylyl cyclase to be activated, or another name, adenyl cyclase. Then cyclic AMP or CAMP is made. And this is our second messenger. So cyclic AMP will activate the kinase protein, leading to enzyme cascade reaction to amplify the signal. Because every time there's a reaction by the enzyme, more products will be formed. So amplify the signal. Kinase enzyme's function is like a phosphorylase. It will phosphorylate the substrate. Kinase function is to add phosphate to a substrate. Okay. Glycogen will eventually be broken down to glucose. This is called glycogenolysis. Glycogenolysis. Okay, lysis of glycogen. Then glucose will diffuse out of the cell into the bloodstream to increase the blood glucose concentration. Okay. Any more? That's all. So total. Cannot. Quite easy, right? Quite easy, provided you study. Okay, what is last question? I'll give you the last game. Explain the role of auxin in cell elongation. Okay, auxin. Any aid from here? Auxin bind to the receptor in the cell surface membrane. It will cause a proton pump to be activated. Proton will be pumped out of the cell into the spaces in the cell wall. Cell wall becomes more acidic or reduced in pH. Therefore, expansin protein will be activated. So expansin's function is to break the bonds between the cellulose molecules, which is the microfibrils. K plus then enters the cell. Therefore, water potential in the cell will decrease. More water will enter the cell by osmosis. Increase in turgor pressure in the cell. So this entire process is called acid growth hypothesis. Okay.